Well, friends, now in the next half an hour, we will discuss another very important aspect of food reactions which normally occur in starch containing foods, and those are gelatinization and retrogradation processes. Starch is a polysaccharide containing glucose units. It is a predominant food reserve substance in plant, and it provides about 75 to 80% of the calories in the food. Food starches are mixture of two structurally distinct components, and those are amylose and amylopectin. Cereal starches on an average contain about 75% amylose and 25% amylopectin. Amylose are a linear chain of repeating maltose unit where glucose units are joined by alpha 1,4 glycosidic linkages. On the other hand, amylopectin molecules are branched chain. In this, both the alpha 1,4 glycosidic linkage as well as alpha 1,6 glycosidic linkages are present. Individual chains are joined with alpha 1,4 linkage, whereas the branches are by the alpha 1,6 linkages. If we compare between the properties of amylose and amylopectin, we see that molecular weight of amylose ranges somewhat in between 50,000 to 200,000, whereas that of amylopectin ranges from 1 to several millions. As I already told you, the glycosidic linkage is in amylase is mainly alpha 1,4, whereas in the amylopectin it is both alpha D1,6 and alpha D1,4 linkage. The molecular shape of amylose is essentially linear, whereas amylopectin are bush shaped. It has branchings. Amylose has high susceptibility to retrogradation, whereas amylopectin have comparatively low susceptibility to retrogradation. Action of beta amylose releases maltose from the amylopectin as well as beta limit dextrin, but from the amylose it gives only maltose. Product of action of glucoamylose or D-glucose in both the cases. The starch granule, as you could see in this image, are made up of amylose and amylopectin molecules which are arranged radially. They contain both crystalline as well as non-crystalline regions in alternating layers. Undamaged starch granules are insoluble in cold water, but they can imbibe water reversibly. That is, they can absorb water, but upon drying they further shrink and come to their original stage. When heated with water, starch granules undergo a process that is known as gelatinization. They absorb water and swell. So, the gelatinization can be defined a process in which the starch granules, when heated in water, swell until ultimately they rupture, collapse and yield a paste. In other words, gelatinization is the disruption of molecular order within the granules. Here you see in the picture, that is, these are the starch granules which are present in the intact grains or in other materials, but when they are soaked, when the grain is soaked in the water, water penetrates, fills up the micellar region and the starch granule swells or grain swells, and upon heating it results into the formation of some sorts of gel. And the temperature at which the granules begin to swell rapidly and lose their property of birefringence is called gelatinization temperature. However, the gelatinization temperature is not a single temperature, it is a range of temperature and it is characteristic of the materials. Right. Cereal starches under normal conditions have a moisture content of about 12 to 14%. The grain, when they are soaked into water, if sufficient amount of water is present, the cold water penetrate the amorphous regions of the starch without disturbing the micelles, that is important, and a maximum water content of up to 30% can be reached within the grain. But what happens when this material is heated, that is, if you provide heat to the water, then the intermolecular or intramolecular hydrogen bonds are broken. This moisture enters into the micellar regions of the grain, that is, grain will absorb more water and swell, and this swelling causes the loss of birefringence. In this figure, you can see the gelatinization temperature ranges of different materials, and this is specific to specific starches, a specific type of starches, and sometimes the gelatinization temperature range is used to characterize the source of starch. For example, you can see here sweet potato, the gelatinization temperature is in the range of 82 to 83 degree. That means it has a very narrow gelatinization range. Its swelling starts at 82 degrees Celsius and it completes gelatinization complete at 83 degrees Celsius. Whereas the starches like rice etc., the gelatinization starts as at 65 degree and it completes, it takes little longer temperature to complete the gelatinization process. It completes at 73 degrees Celsius. As the temperature of the starch suspension increases above the gelatinization range, if sufficient water is present, the granule continue to swell, and this additional swelling increases the viscosity as swollen granules begin to collide frequently. In some cases, the fragile granules may be torn into the fragments, and which results in reduction in the viscosity. And this, whether the viscosity will increase or the viscosity will decrease, will depend upon temperature, initial concentration of the starch suspension, size of the granules, and internal forces holding the molecules together within the granules. In this slide, we have provided, you can see the effect of, that is, the temperature on the viscosity changes in the material. In other words, that is the how that is changing temperature causes the gelatinization, and what is the gelatinization effect on the viscosity and other changes in the material. You could see here that in this range, this is the when you increase the temperature, in most of the cases in the last slide you could see that somewhere in some material it is started at 16, it may in some other material it may start at 65 degrees Celsius, but there is a temperature which says that the start of gelatinization process, that is called TP, that is the pasting temperature. Further increase in the temperature, as I told you that if sufficient water is present, it results into the increase in the viscosity, right, that is hydration of the starch granules take place, and finally it reaches to 0.3, that is which has maximum intensity of gelatinization, and this viscosity at this point is known as peak viscosity, PV. Further increases the temperature, if sufficient water is present, then there is a stage 4 you can see here, that is the complete dispersion, that is enzymatic and shear destruction of the starch granules takes place, and finally the viscosity comes to its minimum point, right, and there is a loss of viscosity at this point, that the temperature is lowered down, right. From this point, if the temperature is reduced, the recrystallization of starch or retrogradation of starch take place, and finally it reaches to the final viscosity 0.7, and then further cooling results into the paste hardening. So, this is the process, that is the gelatinization accounts for the majority of the structural changes of starch granules that take place during heating of a system with specific starch to water ratio. The same thing you can further relate, that it can be taken further discussed with the pasting curve of the starch, that is here in this graph, that is PT point, it is the peak viscosity, that is it is the highest apparent viscosity obtained during pasting, and the temperature corresponding to this point is called peak viscosity temperature. This is followed by a whole, that is ease of cooking, it is indicated by the apparent viscosity in relation to the peak viscosity. Then it is followed by, that is during the cooking, the paste stability is related to or is indicated by resistance to breakdown, which is indicated by apparent viscosity after cooking for a period of 20 to 60 minutes at 95 degrees Celsius. The apparent viscosity of the paste after programmed cooling to 50 degrees Celsius is indicative of retrogradation tendency of the starch. Finally, the apparent viscosity after stirring at 50 degrees Celsius periods for up to 1 hour gives the total setback. So, in this way how we can see what are the from the rheometer, another related instrument, that is we take, that is the to find out the pasting curve characteristics of the starch, and it indicates that is how this starch or gelatinized starch will behave under various processing conditions in the instrument. In this one, it is a figure we have taken another case study showing, that is a phase diagram showing the state and phase transition of starch when applying a temperature profile. In this y axis there is a temperature rise, right, and on the x axis it is the time of processing and storage, right. Initially you can see how what are the with the increase in the temperature and with the duration of the processing like cooking and other process, when heat is applied, how the native starch is changed into the gelatinized starch, all right, and it said come to a stage, that is glass transition, and where this is the, it is a glass transition temperature in which it is in the semi crystalline form. Initially it is crystalline, but with the passage of the time and temperature, this starch structure changes, it becomes to the amorphous stage at this region. And after that, if even further, if it is given further time, then there may be complexion, gelation processes or even the recrystallization process, and the recrystallization process are when it cool the amylopectin normally complexes are retrograde easily than that of the amylose complexes easily than that of the amylopectin. There is recrystallization of the amylopectin normally takes place in the during the storage, whereas retrogradation of the amylose normally takes place during cooling processes. And as you have seen in the earlier figures, when a starch thickened mixture is allowed to cool without stirring or undisturbed, there is a tendency of intramolecular bonds to form, resulting in the formation of a more or less rigid gel. And this is because the hydrogen bonding between the molecules become more extensive, thereby increasing the micellar region, due to which gels become more firm. And when amylopectin molecules are present in comparatively higher proportion, as in the case of waxy starch, the branchage prevents the degree of reformation of the hydrogen bonds, and therefore, it results generally into a weak gel. This is a schematic representation of the changes that occur in a starch water mixture during heating, cooling and storage. You can see here that is this one, it is the native starch granules. When it is heated in water, the granules absorb water and it becomes swollen, it swells. It is followed by further heating is continued to the second B, amylose leaching and partial granule disruption, which results into the pasting of the starch. Further, after it takes gelatinization and then formation of a ordered or crystalline amylose molecule during the cooling process, and ultimately finally during storage there is formation of ordered or crystalline amylopectin molecules during storage. So, this shows how the structure formation and how during the starchy materials, when they are processed, heated, then cooled, then what are the different changes that take place into the starch or in the grains, and which ultimately affects the texture and other characteristics of the material. It may become hard, and that is what you can usually visualize. A simple example, you can take rice. When the rice is cooked, all right, after cooking, when it is cooled down, undisturbed, or even the type of the rice, if it is a stored rice, if it is a waxy rice, then you can get a different types of texture in the cooked rice. That is, somewhere you can get a soft rice and somewhere little hard rice, depending upon the starches and the changes that has occurred during this process of cooking. The another important phenomena is the retrogradation, which you could see in the earlier slides also. That is how the gelatin, after the gelatinization, these swollen starch molecules, when they are cooled, then there is a reassociation of amylopectin or amylose molecule. Normally, amylose molecules try to reoriented parallel among each other to the, and in this, to the extent that affinity for water molecule decreases and the molecule normally precipitates and yields a paste. Okay. So, you can say that starch retrogradation refers to the reassociation. It refers to the reassociation or the recrystallization of the polysaccharide in gelatinized starch, that is amylose and amylopectin. It occurs more rapidly with amylose than with the amylopectin, since amylose is a smaller unbranched molecule. Retrogradation of starch is affected by storage temperature, compositions such as water content, sugar, lipid, salt and anti-scaling agents present in the system. Water content in the material affects the starch retrogradation process. Okay. Crystallization occurs in gel with starch content of up to 50 to 60%. Recrystallization increases with increasing water content up to 40 to 45%, which is result depression of the Tg below room temperature. Further increment in water decreases the recrystallization, and this may be due to excess dilution. Similarly, storage of starch gel at lower temperature, but above the glass transition temperature increases the retrogradation. Storage at freeze temperature below Tg inhibits recrystallization. Higher temperature above 32 to 40 degrees Celsius reduces the retrogradation phenomena. Amylose lipid complex interferes the recrystallization process of amylopectin, which retard retrogradation. Lipids also interact with amylopectin fraction to a small extent, causing formation of amylopectin lipid complex that retard retrogradation process. Even the source of the material, that is the structural difference found in the amylopectin chain of starches from the various sources, also cause differences in the rate of crystallization processes. The short chain of the amylopectin have been found to be responsible for the crystallization of amylopectin molecule, although chains with less than 15 glucose units do not take part. Higher temperature above 32 to 40 degrees Celsius reduces the retrogradation process. In this slide, morphological changes and histological structural changes in the rice grain when it is soaked for 30 minutes at 30 degrees Celsius, and then after soaking it is cooked from 50 to 100 degrees Celsius for 35 minutes in rice cookers. You can see that how the changes takes place with the increase in the cooking temperature process. In the initial soaking stage, the cracks are generated, which you can see here in the pictures. Then these generated cracks facilitate penetration of water from the surface of the grain to starch, which is surrounded by the cell wall. Moisture content increases in grains, and this increase in the moisture content was generated by the starch gelatinization process as well as water penetration into the grain. The gelatinization occurred at a surface earlier than in the interior, and the starch gelatinization between 85 and 100 degrees Celsius, which you can see in these pictures, that is changed the grain shape. It results into the disruption and swelling of the cell wall along the grain surface and caused internal voids. In this slide, you can see that how during extrusion processes, that is extrusion of starch containing material, how the gelatinization takes place and which ultimately results into the decomposition of the or trihydrolysis of the starch molecule into dextrinization or dextrins, etc. The combination of barrel temperature and specific mechanical energy input combined with moisture influences the starch gelatinization process during extrusion. High temperature and extruder speed with low moisture causes structural breakdown and starch damage results due to shear force, etc. Then starch granules results into the gelatinization and then starch is finally damaged, and then this damaged starch may break down into intermediate molecules, that is the dextrins, which have properties intermediate from glucose and starch. There is high screw speed imparts frictional energy in addition to heating energy input. That is, here in this case you can see the shear, heat and moisture increase, how the changes in the rye starch takes into gelatinized starch, and finally there is the free polymers or even dextrinized starch or oligosaccharides and sugar formation ultimately may increase. That all depends upon what are the even temperature and other conditions inside the screw barrel. During the parboiling process, another experiment was reported in the literature, that is the degree of starch gelatinization in rough rice during different soaking temperatures. Okay. And here you see that as the temperature increases, the rate of gelatinization, that is the gelatinization percentage, increases with increase in the time. That is both the time as well as temperature of parboiling process, maybe soaking and heating process in the parboiling results has significant effect on the gelatinization process. Degree of starch gelatinization is extremely dependent on the soaking temperature. It completes at a faster rate at higher soaking temperature than at lower temperature, and the larger duration of soaking at lower temperature also resulted in partial gelatinization of the rough rice. In this, it is a table. It reports result of a study conducted by Yu et al., wherein they have shown the role of starch amylose and its retrogradation, and different rice varieties, maybe waxy rice, very low amylose containing rice, low amylose containing rice, intermediate amylose containing rice and high amylose containing rice varieties were studied, and it was found that most recrystallization within cooked rice occurred during the first 3 days. That is, experiments were conducted, that is up to 14 days. So, the first 3 days, that is the most of the recrystallization process occurred, and which was followed by slower retrogradation, that is, and it ends after the 14 days of the storage. The cooked rice with high amylose content is easy to retrograde, whereas the cooked rice with low amylose content retrograded slowly. Retrogradation of corn starch during storage can be seen in these figures. These are the DSC thermographs of waxy and normal corn starches. The A is the retrograded at 4 degrees Celsius for 2 and 14 days, whereas the B is 1 and 7 temperature cycles of 4 degree for 1 day and 30 degrees Celsius for 1 day, where various starch concentrate. You can see here, that is what is the effect of cycling and starch concentration on the retrogradation of the waxy and normal starches. The shapes of these retrogradation endotherms varied as a function of starch concentration. Narrower endotherm peak were found, that is the paste stored under 4 by 30 degrees Celsius temperature cycle indicated homogeneous crystalline formation than those stored at 4 degrees. The endothermic peak for melting of the retrograded starch became larger with the increase in concentration. This is again another similar study showing the retrogradation of potato starch during storage, X-ray diffraction pattern of potato starch. 3 days retrogradation was enough to form the most aggregation of amylose and amylopectin, which was indicated by this study. However, reheating at 90 degrees Celsius disrupted the aggregated structure of melted amylose and amylopectin as revealed by the relative crystallinities. Reheating of retrograded starch made the crystallinity similar to the freshly cooked samples. The longer cooling and storage time allowed gelatinized starch to aggregate and realign. And this experiment was conducted where these fresh potato tubers were cooked at 90 degrees Celsius and retrograded in refrigerator at 4 degrees Celsius for 1, 3 and 7 days. So, friends, you could see now that how that the important processes, retrogradation and gelatinization processes influence the characteristics of the starch containing materials, and these characteristics finally affect to a great extent to their eating qualities or to their functionalities during food processing and preparation. Thank you.